Okay, so today we are on Wednesday, second day of the week for the week September 21st to 24th. So we're going to try to finish up the muscular system. If we don't, we'll finish it Monday and start nervous system. Next week you have an exam over chapters 4 through 6. You'll need to use honor lock when you do that. So just make sure you've got access to a webcam and that it works. In the event you need it, the testing center is open by appointment, so you need to go to the testing center and schedule an appointment to take the test if you need to do that, but we can't go as a class anymore. So just want to make that available to you. All right. So let's pick up where we left off in chapter 6. Yeah, we talked about the neuromuscular junction last time, and synaptic cleft. All right. So transmission of the nerve impulse to muscle. So again, you have a nerve and it has an impulse, and that impulse gets to the ending of the axon terminal of the motor neuron. The motor neuron is basically just a term that means a neuron that innervates a muscle. Okay. So what happens at that point? So the axon terminal, or the axon, has calcium channels. Remember we learned about channel proteins a couple chapters ago? So channels for calcium will open up and calcium ions will start entering into the axon terminal. So at that point, calcium ion entry is going to lead to the synaptic vesicles. Remember vesicles? Okay. And they'll release a neurotransmitter called acetylcholine, abbreviated ACH. And we'll see a picture of this in a moment. So ACH is going to diffuse across the synaptic cleft, which basically means, remember diffusion, okay? It's going from area of high concentration to area of low concentration. So basically, it's being released by those vesicles into the cleft, and then the ACH will attach to receptors on the cell membrane of the muscle cell. Remember, the cell membrane of a muscle cell is called sarcolemma. So then what? So if you get enough acetylcholine that gets released, that sarcolemma, the cell membrane, will become more permeable to sodium ions for a moment. It's not permanent, but for a while. So at that point, sodium ions can go into the cell, the muscle cell, and potassium is going to leave the cell. Okay, and that creates a difference in charge, and that leads to something called depolarization. And at depolarization, you're going to get opening of more sodium channels. And that just leads to even more sodium ions getting into the cell. And depolarization is going to result in an action potential. And an action potential is needed for contraction. So once you start the action potential, you can't stop it, okay? So basically you have enough sodium ions going into the cell to get a depolarization, or you don't, okay? And then muscle contraction happens. Now, if there wasn't a way to stop this, it would go on and on forever, okay? Your muscles would just contract permanently. So fortunately, your body has an enzyme called acetylcholine esterase. Okay, remember ASE is an enzyme, and its job is to break down acetylcholine. And what does it break it down into? It breaks it down into two parts: acetic acid and choline. So by doing so, by breaking down the acetylcholine acetylcholine esterase will end muscle contraction. All right, so the cell essentially becomes excited at a point of depolarization. So how does it go back to its resting state? Okay, that muscle cell. Well, if potassium ions start diffusing out of the cell, that can help. Also, the sodium-potassium pump starts moving sodium and potassium ions back to their original positions or original concentrations. So remember we learned about sodium-potassium pump a couple chapters back, dealt with that active transport, okay? So everything builds, okay? All right, so this is a picture view of the action potential. So the action potential, it's going to reach that axon terminal, what we just talked about. Okay, that signal. Then, once that happens, the calcium channels open and calcium goes into the axon terminal. That calcium ion entry is going to lead to these synaptic vesicles here, releasing their neurotransmitter, acetylcholine, into the synaptic cleft, that space, via exocytosis. Remember that? Exo out of the cell. Then acetylcholine is going to diffuse across the synaptic cleft and bind to these receptors on the sarcolemma, muscle cell membrane. Then acetylcholine or ACH is going to bind and channels will open that'll allow passage of sodium into the muscle fiber and potassium ions out. But more sodium ions are going to enter than potassium ions leaving, and that changes the electrical conditions in the membrane. All right, so if more positive charge is going in than is leaving, you get more positive charge on the inside, okay? And this leads to depolarization, which causes an action potential. And again, this doesn't go on forever. Acetylcholine esterase enzyme attaches, or acetylcholine can attach to acetylcholine esterase and break it down, and it stops muscle contraction by closing these channels. And the ions can no longer get through. So sometimes it helps to compare things you don't understand to things you do understand. So let's compare the action potential to a flame consuming a dry twig. So if you've ever, if you had a burn pile, or if you, burn things in chimney when it gets cold, you've seen this, you see a small twig and it starts, the flame ignites it, and slowly that flame starts spreading along the twig. So this is basically how a nerve works. The stimulus is going to make that action potential travel along that nerve. Then you get diffusing of sodium into the cell. Then you lead to the action potential. And it starts spreading across that muscle, just like that flame spreads across the twig, okay? All right, so let's take a break here. Let's talk with your partners about what you learned about how muscle contraction works and the role of acetylcholine and acetylcholine esterase. And you can do this at home as well if you're watching at home. All right, let's talk a little bit about sliding filament theory, okay? It has to do with how muscles contract. So if you remember last class, I believe we talked a little bit about actin and myosin, about the thin filaments and the thick filaments, right? So calcium will bind to proteins on the thin filaments on the actin. It'll expose binding sites on myosin, okay? Because they kind of sit on top of one another. And so by binding to the actin, it moves the actin over so the myosin's free. It allows those myosin heads on those thick filaments to attach. And those attached heads are going to then pivot or move, and it slides those thin filaments toward the center of the sarcomere. Remember, we learned about that too last class. And that's what leads to muscle contraction. So this is happening at the same time you get the depolarization, okay? All right. Because the depolarization is really a way of describing your muscles from a kind of like an electricity standpoint. This is more describing it from kind of a physiology standpoint of the pieces. All right. ATP is your energy source for the sliding. And you'll continue to have that ATP as long as you have calcium ions. All right, so you should hopefully remember at least looking at something that looked like this one on the top last class. So this is a sarcomere. It has those thick filaments in red in the middle and the thin filaments in blue on the sides. Remember, that's the unit of muscle contraction. This is in a relaxed state, okay? And you can tell it's in a relaxed state because you have a large H zone, okay? Area where you just see thick filaments. So when a cell is fully, or a muscle cell is fully contracted, you have a very small or almost non-existent H zone, okay? Because the thin filaments have slid toward the middle of the sarcomere. All right, so what does that look like in a little bit higher magnification? So in a relaxed muscle cell, the regulatory proteins will form part of your actin myofilament, and that prevents the myosin from binding. And so when you get an action potential, the muscle cell gets excited, the calcium ions again get released from the areas where there's storage. And these, the area where you store your calcium ions are your sarcoplasmic reticulum, the muscle version of endoplasmic, of smooth endoplasmic reticulum. Okay, it stores up the calcium and it's released whenever there's an action potential. All right, so that again, that flood of calcium is a trigger for contraction to happen. And that calcium ions are going to start binding to these proteins on the thin filaments up here. And those proteins will change their shape and their position at that point. And this exposes binding sites on the myosin, the thick filament, and allows the myosin heads to attach to the actin. And essentially it's pulling it toward the middle. Okay, it's like it grabs it, moves it in. So when these myosin heads get free, they're in what we call a cock position. And the myosin attachment to the actin, the thin filament, works like a spring. And it causes those myosin heads to snap or pivot toward the center of the sarcomere. And when that happens, thin filaments get pulled toward the center. Okay, so it's like if I'm holding something up here and I am attached, if I move toward the center, what I'm holding will also move toward the center. Okay. So how does this work? Well, ATP gives you energy in order to do this. And every time you move, basically recock the myosin head, you need ATP. And then once the action potential is over, calcium ions get returned back to the sarcoplasmic reticulum. And those proteins move back to their original shape, original position, allows your muscle to relax. And it goes back to its original position. And those myosin binding sites are blocked again. All right, so give you a minute, talk with your partner about sliding filament theory before we go on. All right, so let's talk about muscle contraction a little more and skeletal muscle. So the contraction is said to be all or none. Either you get contraction or you don't. But within a skeletal muscle, some fibers can be stimulated in the same interval, but others may not. And so having different combinations of muscle fiber contractions gives you different responses, and we'll talk about what that looks like. So you can say the responses are graded, which means you have different degrees to which the skeletal muscle actually shortens. So how does this happen? So if you change the frequency that the muscle is stimulated, or the number of muscle cells that get stimulated at any given time, you can change essentially the amount of contraction you get, right? So if you stimulate one muscle cell, you'll get a certain amount of contraction, but if you stimulate 10, you're gonna get more contraction. If you stimulate it once every second, you get some contraction, but if you're stimulating 10 times a second, you get more contraction, okay? So we have different terms we use to describe these graded responses. One is twitch, and that just means you have one single brief contraction. Okay, so you may have seen someone have a twitch in their muscles. It's just single and brief. It's not considered normal function. And if you look at it on a graph, look at the amount of tension or force that contraction generates in response to a stimulus, will look kind of like this over time. Okay. So depending on the strength of that stimulus, you'll get a certain amount of contraction. Now, there's another thing that we learned about called summing of contractions. And that just means that if you have one muscle contraction and you cause that muscle to contract immediately following that. And so when you get more frequent contractions, your muscle doesn't get a chance to fully relax, okay? And because it doesn't get a chance to fully relax, you get a summed contraction. So you get more contraction than you do for that individual contraction. So
maybe like if you're eye jumping or like a leg cramp?
So essentially, if I would say more like if you have a sudden eye movement, that's more like a twitch. So cramp is a little bit different. Because in that sense, it's not relaxing. So here's the summing of contractions. So you have a stimulus, you get a certain response, starts to relax, but you don't let it fully relax. And then you give a stimulus again, you get even more tension, even stronger contraction the next time. Yes?
So that would coincide with like a leg cramp, because sometimes it starts easing up and then it comes right back. So
yeah. So if you get if your nerves are sending a signal to your muscles to contract before it has enough time to fully relax, you would get a summed response. So in a sense, yes. So if you, but, and we'll show you even more of a reaction that you'll see that looks kind of more like what you're describing.
So is this kind of like as like if you're cold and all of a sudden you get a chill and you get a little like a little tremor? Is that what
It's similar, yes. So yeah, because if it's not something that you're doing repeatedly, yeah. But it's your body sending a signal to your, or your brain sending a signal to your body that you need to warm up. All right, so we also have tetanus, and then we're not talking about the disease tetanus. This is actually a term we use to describe muscle contraction in this case. And it can be incomplete or unfused. That's one form. In here, you get some relaxation between the contractions, but you're getting nerve stimuli at an even faster rate than we saw during summation. All right, and so unless the contraction is smooth and sustained, we call this unfused or incomplete tetanus. And so this is what that looks like. So again, this is more rapid and than what you would get with summation, but you get even more, even stronger of a contraction. Okay. It can be. It can be in terms of the muscle activity. The other piece of this that I want you to know is you do get some relaxation. You can see you do get peaks and valleys. Okay, so each peak and each valley is higher than the one before it, up until you get to a certain maximum tension, but you get this sort of stair-step pattern. Okay, not to be confused with the fuse or complete tetanus. And here you get no relaxation. You keep stimulating that muscle so quickly, it has no time to relax. Okay, and you get a smooth and sustained muscle contraction. So here's the difference. Because those stimuli are coming so fast, it's smooth. You never get any peaks and valleys. Okay. Now, you may get to a certain point where the amount of increase in tension slows down, like it does here compared to here. Okay, but it's still smooth. Okay. So that's the difference between incomplete and complete tetanus. So what I really want you to be able to do is to be able, like if I were to give you a question to ask you, to identify which one, if I told you, hey, this one, you get increased stimulus, but you have some relaxation, that, you'd be able to tell me that's incomplete tetanus. But if I said you get increased tension, but no relaxation, that's complete. Alternatively, if you're able to look at a picture and tell me which one of that, is it a twitch? Is it summation? Is it unfused tetanus? Is it fused tetanus? But here we're talking 4 pictures. We're not talking 30 pictures. You gotta be able to tell the difference there. Okay. All right. So let's take a break here. I want you to explain your own words: twitch, summation of response, unfused tetanus, and fused tetanus to your partners. Y'all can. Yeah. All right, so let's talk about muscle responding to stimuli. So the force that you get out of any muscle will be consistent with how many fibers you stimulated. Few fibers, small force. Lots of fibers, lots of force. All right. So it's directly proportional, if that makes sense. Again, if you get more fibers involved, you get more fibers contracting, you get more tension. And muscles will continue to contract until they run out of energy. That should make sense. I mean, I mean, I don't know how many of you either weight train or do push-ups, sit-ups, any of that kind of stuff. You know, you're able to do them until your body gets tired, right? And you're tired because your ability to get ATP to your muscles is compromised in terms of your breathing or just your level of fitness. Your muscles haven't adapted, right? So where does the energy come from? ATP, right? Adenosine triphosphate. So where do you store it? It gets stored in your muscle fibers in small amounts. They're used up, so you don't really store a whole lot. You store some, but not a lot. So you have some just to get you going, but you need ability to be able to make it when you need it. So how do you generate ATP? You can phosphorylate ADP, and that basically is a fancy word that means you take ADP, which has 2 phosphates on it, you add a phosphate group, okay? And you take that phosphate from creatine phosphate in order to do that. You also have aerobic respiration, which basically aerobic means needs oxygen. So it's breathing, you know, you think respiration, you think breathing. Here we're talking really about that's, you know, breathing provides oxygen so that you can create ATP. And the chemical reaction, so here we're talking about the chemical reactions that lead to making ATP that involve oxygen, okay? Now, making ATP without oxygen is called anaerobic metabolism. And so that process, those processes that are anaerobic are glycolysis and lactic acid formation. Okay, so you can get some ATP from glycolysis, not much, but it doesn't require oxygen. But if you don't get oxygen, you can't do aerobic respiration. All right, so just talked about this. Fastest way to get ATP is take a phosphate from creatine phosphate to make ATP from ADP. And your muscle cells do store creatine phosphate, okay? And it's considered a high energy molecule, okay? So, you know, that's part of the reason why, you know, people who do a lot of bodybuilding and all take a lot of creatine because it's able to help them have energy for their muscle contractions. So ATP, once it gets depleted, you end up with ADP. And the creatine phosphate will transfer a phosphate group over to ADP, make ATP. So once you exhaust all of your creatine phosphate, you know, that happens in around 15 seconds. So it doesn't take too long to get rid of all of your creatine phosphate. Now, for every creatine phosphate, you can get 1 ATP because there's 1 phosphate, right? So it's fast, but it doesn't, you know, it's not getting you all the ATP you're going to need. And it's fast because there's not a lot of steps involved. Okay, so here's another view of that direct phosphorylation. So again, you take your creatine phosphate and your ADP, you move that phosphate over here, you get creatine and ATP. Doesn't require oxygen, you get 1 ATP for every creatine phosphate. It takes about 15 seconds. Now for aerobic respiration, you're taking glucose, sugar, breaking it down, you make carbon dioxide and water from it. Okay, but you get lots of ATP in comparison. You get 32 molecules of ATP. And it happens through a series of reactions or metabolic pathways in your mitochondria. Remember those? Remember we said that the mitochondria is your powerhouse of the cell, makes the ATP. And it's a much slower reaction because there's lots of steps involved. And you need lots of oxygen for this. And your products, again, water and carbon dioxide. Okay, so here's your, again, aerobic cellular respiration. Then you have your glucose. So it can be directly in your blood, or you can break down chains of glycogen to get the glucose molecules. Okay, it can also involve fatty acids or amino acids. And just to make the pyruvic acid, you don't need oxygen, but to take the pyruvic acid in your mitochondria and make ATP, you do need oxygen. And as byproducts, you get carbon dioxide and water. So oxygen overall is required. You get 32 molecules of ATP, carbon dioxide, and water. But this takes hours for every glucose molecule. So if you eat something, you know, you're not necessarily going to eat something and then right after have enough energy that you need to go run a marathon. I mean, you kind of have had to get your stores of glucose. You know, you have to have that in place well before. All right. So there's also anaerobic processes, glycolysis and lactic acid formation. So you're breaking down glucose with no oxygen. You get 2 pyruvic acids, okay? So essentially glucose has 6 carbons in it. Pyruvic acid has 3 carbons. So you just basically cut it in half. And you get 2 ATP molecules out of that. And that pyruvic acid, then you can take that and make lactic acid. Okay, it's fast but inefficient in the sense that you don't get a lot of ATP. So you need lots of glucose to get the ATP you need for this process. But the problem is you get lactic acid. And what does lactic acid do? Makes your muscles hurt, right? Makes your muscles tired. All right, so here's a visual of that. Again, take your glucose, go through glycolysis in your cytosol, make ATP, pyruvic acid, take the pyruvic acid, make lactic acid. All right, so then you're making 2 ATP for every glucose molecule. It takes about 40 seconds. All right. So let's take a break here, talk about aerobic metabolism, anaerobic metabolism. Okay. Similarities and differences. So let's talk about muscle fatigue. So if you get strenuous muscle activity for long periods of time, your muscles are going to fatigue. And you end up, that's as a result of imbalances in your ions. You get accumulation of lactic acid in your muscles, and you get decreases in your supply of ATP. And so once you finish exercising, you repay the oxygen deficit, okay? And how do you do that? Breathing deeply, right? When you finish, I'll go out and try to run a few miles in the morning, and then, by the end of it, I'm, breathing pretty heavy. And you think that's just because you're tired, but that's really your body's way of getting oxygen in so you can get your ATP so you can recover. Okay, let's talk a little about different kinds of muscle contractions. All right, so isotonic contractions. So the filaments are able to slide past one another during muscle contraction. So your muscle will shorten, you get movement. So if you bend your knee, right, rotate your arm, these are all isotonic contractions. So if you have an isometric contraction, you get increased tension, but you're not able to shorten your muscle or produce any movement. So if I'm able to move, so I'm moving my leg, you have isotonic contractions that are happening in the muscles. But if I push against the wall, I'm pushing force against the wall. I can feel it in my arm, but there's no movement. So that's isometric. So remember, iso is same, metric is distance, so same distance because there's no distance moved. All right, so muscle tone keeps your muscles healthy, keeps them ready to react. And this happens as a result of nerve impulses that are delivered to the different cells in your muscles. Now, if you damage the muscle supply, your muscle is going to lose muscle tone. And it can get paralyzed and atrophy. Okay, so it's important to make sure so if you injure yourself, it's not just necessarily that you injured the muscle, you could injure the nerve that supplies that muscle. And then as a result, the muscle is going to have some problems. So benefits of exercise, you can increase the size of your muscles with exercise. Depending on the kind of muscle or type of exercise you're doing, you can increase the strength of them. You can increase their endurance. So in some cases, you can make them stronger or bigger. In other cases, you can just make them able to work for longer periods of time. So again, push-ups as an example, when I started trying to do push-ups in mornings or whatever, I do a few push-ups, but now I can do more push-ups, right? Not necessarily my muscles aren't bigger, but they have more endurance. So aerobic endurance exercise, running, jogging, biking, you get longer and more flexible muscles that are more resistant to fatigue. So like some people are really good sprinters and other people are really good distance runners, right? You said you used to do mile relays, right? So it takes a different kind of running to run 100 versus running a 400 or 1600, right? So it's very different. And so whatever you're good at, you have different kind of muscle fibers, right? So say, someone who's like Usain Bolt may have muscle fibers that's a high concentration of certain kinds of muscle fibers. And then someone who runs, 4 miles, that runs a mile in 4 minutes, they have different concentrations of muscle fibers, okay? Just because they run very different kinds of races, right? So your body metabolism becomes overall more efficient when you exercise. You have better digestion, better coordination. Your body gets more out of the food that it eats. So with resistance training, more and also more isometric type exercises, you get more size and strength. Okay, so it just depends on which type of exercise you're doing. So here's more of your aerobic exercise. This is more of your isometric or strength training, right? So depending on which type of exercise you're doing, your muscles will adapt to that. Okay. All right, so we've got 5 golden rules of skeletal muscle activity. So hopefully this helps you kind of think about things, the big picture. So for the most part, All skeletal muscles are going to cross at least one joint. And typically the bulk of your skeletal muscle is going to lie close or proximal to that joint that it crosses. So all skeletal muscles are going to have at least two attachments. They'll have an origin and they'll have an insertion. Okay, they'll usually attach to two places. Skeletal muscles only pull. Okay, they don't push. So we might think of ourselves, you know, like if you're doing push-ups or, you know, bench pressing, you think of yourself as pushing, but in reality, you're actually, when muscles contract, it's because the muscle is actually pulling. Okay, because again, you got an origin and insertion. So even though it may look like I'm pushing, right? So what's actually happening here on my tricep is that my tricep is actually contracting. It's actually pulling, okay? So it's pulling to make the muscle contract. All right, so during contraction, the skeletal muscle insertion moves toward the origin, okay? All right, so this is chapter 6.